Okay, so homework time again. Highlighter, pen, pencil, ruler, calculator. I don't know if you're getting a grip on what you need now after all these videos. So, okay, so what have we got here? We've got a photograph and we've got a lovely diagram, and this is a photograph, looks like taken down a microscope. So, the photograph below is taken using a light microscope. Good guess. Under high power. It shows part of a villus from the small intestine. Okay, so what do I know about a villus? It looks like that. It's got a layer of cells here, which are the epithelium cells, and every so often we've got a little gobletty cell. And then in the middle we've got a lacteal, and we've got capillaries, and here we've got the lumen. So I need to sort of transpose what I know, the diagram, to what I'm seeing here. So white gap, I'm thinking that will be the lumen. I think so. These will be nuclei here. So I'm thinking that if I'm next to the lumen, I'm thinking what I'm looking at are these epithelial cells, and then these black blobs here, they look like they might be goblet cells. And I've got some folds, and I've got that looks like a brush border, maybe? No, brush border. So that's the microvilli. And then I've got this sort of gap in the middle, that might be a lacteal there, or maybe a capillary or something. Okay, so off we go. Let's see, what questions are they going to ask? Identify tissue A. Right, identify cell B. Okay. So, tissue A, that's this layer here. So that I know is epithelial tissue. Identify cell B, and I thought that might be a goblet cell because it's different from the others. So identify cell B, goblet cell. And it's identify and describe its function. Describe its function. So what do goblet cells do? It's a goblet cell, and its function is to secrete mucus to protect the epithelium from digestive enzymes. Okay, excellent. So a little bit of shift of focus, the diagram opposite. Okay, so that's that diagram over there. There's a cell, one cell in the small intestine showing uptake of glucose from the lumen. So here we're now talking about going across that cell. So what can I see on this? So here I've got my microvilli. There's spilled hair everywhere. Microvilli, and I know that they increase surface area. So what am I seeing now? So I've got cell membrane, I've got my tight junctions there, look, to prevent leakage. And then I've got this feature in here which seems to be transporting glucose from the lumen into the cell, and it's also at the same time transporting sodium ions into the cell, so I'm thinking that that's a protein. I'm not seeing any energy, so I'm thinking that's some kind of passive process. And then down here I've got, oh, these look like 2 more proteins. So the glucose is coming in and then leaving through this protein Z. And then my sodium is coming in passive, and there here I've got ATP, so that's active, and it's leaving by active transport, and it's a sodium potassium pump, which makes me think of nerves, but I don't think that's what's going on here because we're in the small intestine. Okay, so using the information in the diagram above, right, so I've got to use this stuff up here in the diagram above, explain what's occurring at X, Y and Z and how this leads to uptake of glucose. Oh, this is going to be trickier. So my glucose is diffusing, it's being transported with sodium, and the sodium's being transported out, so that's active, and that's going from low to high. So as my sodium's coming in, we're then actively transporting it out, so we've got a low concentration, that will keep that coming, so that's a concentration gradient, because we'll have low there and we'll have high up here. And if it's pulling in glucose with it, then we'd have a high concentration of glucose going out to a low concentration in the blood, and it wouldn't really matter what the concentration of glucose was as long as we keep that concentration of sodium low in the cell. Okay, so that's my thinking on this. So the first one we're doing is X. X is this one at the top. So X, sodium ions and glucose are being co-transported into the cell. No energy, so no energy and a protein carrier means it's going by facilitated diffusion. So that all looks good. Then Y, so Y is this one with ATP, so active transport, and I need to talk about how this leads to uptake. So sodium ions are being actively transported out of the cell and maintaining, I can't spell, maintaining a low concentration inside the cell to keep the co-transport happening. And I think if I was doing this in an exam I would potentially use a bit of extra paper, but I'm not, so there we go. So what's happening at Z? At Z, the glucose, so Z, glucose is being transported, no energy, into blood and capillaries by facilitated diffusion. So just before we leave this question, this is exactly the same process that takes place in selective reabsorption in the kidneys. So glucose is always co-transported with sodium ions, and the sodium ions are always transported by active transport out, which keeps the glucose coming in. So it's exactly the same story as you will encounter when we do selective absorption in the kidneys. So now we've got a big pile of information down here that's going to be then therefore unfamiliar and AO2. Jones disease of cattle is caused by pathogenic bacteria which are taken up by cells lining the ileum. As the body tries to rid itself of these bacteria, the immune response causes a thickening of the tissue lining the intestine, loss of microvilli. This prevents it from functioning normally. Although animals may be eating normally, they gradually begin to lose weight. Using the information above, so I'm going to have to go back to that, and your own biological knowledge, explain weight loss in cattle. Okay, so the gut, so the lining ileum, we're talking about absorption, thickening, we're talking about diffusion paths, loss of microvilli, surface area, and then we need to explain how they lose weight. Okay, so the thicker, what did they say, got thickening, thicker epithelium, so the tissue lining the intestine, lengthens the diffusion path, and loss of microvilli decreases surface area. So the rate of diffusion of nutrients is less. This means less nutrients are absorbed. So now explaining why they lose weight then. What are we looking at? Cattle. Cattle lose weight as they use up body fat to compensate for the less no, that's a rubbish sentence. To compensate for less nutrient absorption. So don't forget that to lose weight you have to use up your body fat. That's the only thing that's ever going to make you lose weight. So that seems to be quite a common theme